Alrighty, so let me first mention that as I advertised earlier, I put our part 2 of test 3 on Moodle for you to enjoy yourself with. So please go out there. It has to do with bonding of atoms together, making molecules, making ionic compounds, and a little bit about the taxonomy of organic molecules in terms of how the name is related to the structure. So make sure you go out there and do the exercises for our second part of test 3. Now remember that the usual deal as we've been doing in the past. I'm pitching in a few points and you guys pitch in your own points, and then we'll see how that goes. All right, so this is open now and will remain open until Monday. I believe that would be the 16th, would it not be? The 3rd, and then the 10th, the 16th of November, Monday at 11:30. That will close. So please go out there and complete the exercises for your points. Alrighty then, what we have done is sort of moved a little past traditional chemists' organic chemistry. Remember, organic chemistry is this chemistry of carbon and the connections that carbon makes. And one of the reasons it was called organic chemistry, or the reason I guess we should say, is that the origins of the word organic have to do with something meaning alive. And of course, as we've said many times over, carbon-based chemistry is the basis of life. All life forms we know of have kind of miraculously, amazingly similar chemical compounds involved in them being alive. I mean, really, it's quite remarkable, even the similarity between many of the compounds that make a tree alive and that would make an animal alive are, again, incredibly similar in many regards. And the foundation, of course, for all these systems is a carbon structure. Now, as I've mentioned several times over, but must mention again, really what you should think of the carbon as is sort of a tree or as I often have said, a closet rod. And then on this closet rod, you hang things. And it's the order in which they're hung, and it's the things which are actually hung, which give these organic molecules their so-called functionality. It makes them operate in a fashion that ultimately leads to an object becoming animate. All right, so with that in mind, I guess what I'm going to do is turn to our slides. What we're beginning to do is just a very brief introduction into some of the molecules that are involved in making something alive. And again, I will state very brief. We have several different classes here in the chemistry department that focus on the molecules that make things alive. Of course, again, much like all our other chemistry classes, they're at various levels of depth. So I would offer to anyone who might have an interest in some just perfunctory or basic knowledge of molecules of life, you could take a rather straightforward class on it, or you could, if you so desired, delve deeply into the subject and learn the nitty-gritty about molecules that keep objects alive. Personally, I am not that particular type of chemist. Chemistry is a really niche science. There's so much breadth of knowledge that goes into chemistry that typically people who are masters of chemistry, I'm a doctor of chemistry, I've actually when you get doctoral degrees in chemistry, you focus on a narrow region of chemistry. Biochemistry is not my particular area of focus, but still I know enough biochemistry to be dangerous. I would say I know enough to certainly discuss things of biochemical interest with us here. All right, so again, let me turn to my present now button, and I'm going to present my stuff and hit the buttons that make it so that this all happens. And what do I want to do? This is the one I want. Yes. Okay, so I'm turning on the show, and where we were was a discussion of molecules that we classify as a carbohydrate. And as I often mention, this is a name that's common not just to people who study chemistry. It's common in the dietary science and in food science and even in general nutrition. And I would go so far as to say, of course, your mom and them have heard the term carbohydrate. And the implication of the name carbohydrate is that somehow it contains carbon and the hydrate implies water. And maybe the origin of this name comes from the fact that the general formula of a carbohydrate is CnH2ON. So if for every N units of carbon in a carbohydrate molecule, there are N units of H2O. Now, they are not hooked together so that a carbon is hooked to a water, carbon hooked to a water, carbon hooked to a water. They are spread in various ways, as we will see in some examples upcoming, spread in various ways throughout the structure of the molecule themselves. Most carbohydrates have as a basis some sort of ring structure, but I will point out to you that not all carbohydrates have a ring structure associated with them. So far and away, the most important of the carbohydrates is something that we call glucose. Glucose, it turns out, is the basic building block of the plant world. Glucose, as it turns out, is the basic fuel of the animal world. So when plants generate glucose, they don't get it naturally out of the ground. Plants synthesize glucose or make glucose. So in a synthesis process or a building up process, as we talked a little bit about classifications of reactions types before, in a synthesis reaction, what plants do is take basic ingredients from the planet and they build with those basic ingredients these glucose molecules. Now, what animals do is we take the glucose molecules and break them down. And when we break them down, we're ripping apart the connections, of course, in this so-called carbohydrate molecule that were put together by the plant world. But most importantly, the point of us breaking down these connections that the plant world built for us is to release energy that is stored in the connections that were made to build the carbohydrate. Okay, so there are these two processes that govern these behaviors, and they are known as photosynthesis and respiration. So in plants, what photosynthesis is that the plant has the ability to take from the surroundings carbon dioxide and water. Now you will notice that for a plant molecule to build a glucose, it has to take 6 carbon dioxides and 6 water molecules. Now, when the plant takes the 6 carbon dioxides and the 6 waters, it rips the carbon dioxides apart, it rips the water molecules apart, puts these things back together, and makes the molecule that you see on the right as a product, or as a matter of fact, makes 2 molecules that you see on the right as a product, as products. And the product molecules that are formed from the process of photosynthesis are C6, H12, O6, and O2. All right, so let's spend a little bit of time discussing this equation, because I can't overemphasize to you the fundamental importance of this equation and what it means for this planet and what goes on this planet. Photosynthesis, you will note, has as reactants CO2, H2O, and energy. Now, the plants generally suck the H2O out of the ground. That doesn't necessarily have to be true. It is possible that the H2O can be absorbed directly on from landing on the plants, but almost entirely the H2O is absorbed by so-called capillary action through the ground system. The polarity of molecules in the cellulose of the plants kind of draws water molecules through the plants, and the water molecules climb through the plants to the factories of photosynthesis. And I think we would be surprised to note that the factories of photosynthesis are the leaves on the plants. So the water makes its way through the root system up to the leaves, and in the leaves it is combined there with oxygen. The leaves absorb oxygen out of the sky, and then they combine the oxygen with the hydrogen. You'll notice in order to do this, an amount of energy must be input. So what we call photosynthesis, therefore, is an endothermic process. Now, as a matter of fact, our next unit of discussion will be on energy, a brief introduction to the chemistry of energy. And when you have an endothermic thing, it requires energy to pull it off. So why does photosynthesis require energy? Well, we have to break apart the CO2 and H2O to recombine to form the glucose and the oxygen. So the energy that is required is energy to drive this process of breaking apart the reactants and building the products. And of course, we're not surprised to learn where that energy comes from. It's not really created magically in the process of photosynthesis. The energy is absorbed by the plants, just like the carbon dioxide and water are absorbed by the plants. And I don't think we would be surprised to learn that that energy comes from sunlight. So green leaves take sunlight, and the green leaves take the sunlight, and then they combine CO2 and H2O, and they form this product glucose and O2. All right, so let's talk a little bit about the ramifications of this equation. Of course, what do plants do with the C6H12O6? Well, they now use it in a structural sense. They use it to form everything that plants need to grow and thrive. Like what? Like more leaves. Like what else? Well, like stems and branches and twigs and things. Everything that is part of a plant system, the structure of the plants, including, of course, the fruits that might hang from the tree, the leaves that hang from the tree, everything has as a structural component this lattice work of glucose molecules. Now, as we will see upcoming here, it is typically not independent glucose molecules that plants use. They begin to combine the glucose molecules together most importantly to make a material known as cellulose, but plants make other materials as well, including a material known as starch. And we will, as we move forward here a little later, we'll investigate some of these combinations that the glucose takes to make these. Now, it not only does that, it makes connections of glucose units to make things like fructose. And what is fructose? This is the thing that when you bite into a fruit, it makes the fruit taste sweet. So it will take some of these things and put them in certain combinations to make these collections that contain their seeds enticing to animals. They want to, one of the clever and cunning things that nature developed were these enticing dangling baubles hanging from the plants that would entice an animal to eat this. And then what the animal would do would be walk away from the tree after it ingested these tasty morsels that have these certain chemicals in them to have flavors and also aromas and of course also sweetness. So the plants enticed the animals to eat them. And the main reason plants enticed the animals to eat these was at the core of these enticing morsels that they were creating for the animals were their seeds. So the plants would entice the animals to eat their seeds surrounded by these coatings of things that the animals liked. So then the animal walks away, maybe a few feet away, but more likely a mile or so away. And then the animal redeposits the remainder of this fruit onto the ground in an excrement. And so this enabled the plants to spread and proliferate. See, there's a competing way in which other simpler forms of plants learn how to spread. And this would be airborne spread. So there were certain plants that made little wingy looking seed pods. And when the wind rippled through the limbs of these typical plants, spores of seeds would float in the wind and get carried out and deposited on the ground. And then that would spread those types of plants. Well, what was quickly discovered, not discovered, but I guess what was adapted in the evolution of plant life, they saw that they didn't reason this out, but the result of this occurrence was that when they got animals involved in carrying their seeds by putting it into some enticing food source, the animals could carry the seeds very far. Remember, trees are sedentary. They don't move. So if they're going to just drop leaves or seeds right off of their limbs, they're going to be growing more trees in a very densely packed region. In order to proliferate and spread, they needed to get some animate object that could be more mobile. And it turned out that animals were a great source of carrying the seeds away. All right, so that was a long story to explain why these certain glucose molecules ended up constructing sugars and ended up constructing fruits and actually other molecules as well to make these enticing morsels for creatures to eat. Let's turn back and look at more important aspects of this equation. Notice that this is a so-called balanced equation. Now, what does it mean that this is a balanced chemical equation? Atoms are not created or destroyed in the process of the combination, the reactants coming apart and the products being formed. If you will notice closely, there are 6 carbon atoms on each side. There are 18 oxygen atoms on each side. Is that right? Yeah, there are 18 of them. And there are 12 hydrogen atoms on each side of the equation. This is the way Mother Nature works. Atoms are not created or destroyed during chemical reactions. The coefficients in front of the molecules then tell you how many of them you simultaneously need to react in order to pull this off so that atoms don't have to be created or destroyed during the process. What it's saying is whenever this reaction occurs, to form one glucose molecule, 6 carbon dioxide molecules and 6 water molecules have to combine in order to make a single glucose. And another byproduct of the formation of this glucose is the release of 6 O2 molecules. Now, I'm sure we're all aware, O2 is the magic ingredient of aerobic creatures. Aerobic creatures are animals that breathe in oxygen to drive their metabolic systems. Not all animals or things on the animal side of the equation really do, but the important ones, the advanced ones, the main, most important of all animal life on this planet lives via aerobic respiration. Aerobic, A-E-R-O-B-I-C respiration means that you use oxygen as the fuel to drive your metabolic process. Okay, so then again, let's turn to the left side of this equation, and let's notice that in the process of performing its photosynthesis, a plant will absorb sunlight through its leaves, the factories of photosynthesis. It will then take 6 carbon dioxide molecules from the air, suck 6 water molecules up through its roots, rip that all apart, recombine them, build these glucose molecules, which again now it can use to make structure for its to grow and expand, and it will also in the process release 6 O2 molecules. Now, the atmosphere on this planet is 20% O2, and I will venture to tell you that not every O2 molecule in our atmosphere was deposited there by plants. Much of it was also deposited by algae, but algae, if you really want to get technical, are slightly different than plants. They do carry out photosynthetic type processes though. But what you should take from this is that if there weren't either algae or plant life on this planet, we would have never got the oxygen in the atmosphere that made it possible for animal life to exist. When this planet was formed, there was not oxygen already naturally in the atmosphere. It took millions of years for the oxygen level to build up to the levels that it has reached so that animal life could exist on this planet. So this photosynthetic reaction, whether again it's being performed by plants or whether it's being performed by animals, is absolutely fundamentally crucial in order for animal life to have evolved and taken over this planet. Okay, now the other thing we should keep in mind is not only is that the case, also notice not only is photosynthesis producing oxygen for us, photosynthesis is also removing carbon dioxide from the atmosphere. So there is a huge amount of carbon dioxide being scrubbed out of the atmosphere, and it's been occurring naturally for millions of years during the photosynthetic process. Had this not been the case, there would have been a massive buildup of carbon dioxide in the atmosphere. And one of the problems that presents, whether you want to believe it or not, is that carbon dioxide is an excellent trap of heat. When the planet gets hot, it radiates the heat back out into space, But when there's a huge amount of carbon dioxide in the atmosphere, instead of being able to radiate out into space, the CO2 naturally traps that heat in the atmosphere, which causes the atmosphere to get a little bit warmer than it would have been had there not been that much CO2 around. Now, don't get me wrong, there's still plenty of CO2 as we're all well aware, and we're producing way more CO2 now than is being scrubbed out of the atmosphere in the photosynthetic process. So we are at an imbalance in terms of our efficiency in the CO2 cycle of this planet. But it's a good thing that plants are very good at scrubbing CO2 out of the atmosphere, or we would have reached the tipping point that humans are trying to send the Earth into well before we are reaching it now. So it's very important that plants are able to remove CO2 from the atmosphere in order to keep the planet from heating up to some ridiculous temperature that is too hot to sustain life anymore. All right, now another thing we should mention before we move away from this equation, we should mention the fact that leaves are the plant, if you like, the synthetic plant part that will take the energy from the sun and then combine it with these two ingredients to make the glucose and the oxygen. But I think it's readily apparent what type of light is used by plants to drive the photosynthetic process. Plant leaves invariably are green. Now let's think back to what we had talked about. Now it seems to be some time ago, but some time ago we had talked about what an object which appears green, why that object appears green, what it is doing to make it appear green. Light of all colors is striking the leaf of a plant. When the light of all colors strikes the leaf of the plant leaf absorbs certain colors. And then it reflects the remaining colors to your eyes, which is what you see. So when all colors of light strike the leaf, after it bounces off the leaf, we see green. What does that mean? Well, if we divide the colors of all into really three primary colors, the reds, yellows, and blues are striking the leaf. The blues and yellows are bouncing off the leaf, coming to your eye. So the green light is coming out, the red light is getting absorbed. So during the course of photosynthesis, plants absorb red visible light, and the molecules in them, the so-called chlorophyll molecule, absorbs red visible light, takes that energy from the red visible light of the sun, and breaks apart the CO2s and H2Os to make the glucose. Now, there's some interesting aspects to that. One of the reasons that nature has settled upon red visible light as the driving force for photosynthesis is because the peak output of radiation from the sun is very close to the red visible light part of the spectrum that is coming off the sun. It's not exactly the peak, but it's really close to it. This was the best molecule that nature could design to take advantage of the maximum amount of energy that it could get from the sun. Now, another reason that red is preferred by leaves is it turns out that the longer the wavelength, which is what red light is, the deeper the radiation can penetrate into the leaf. So red light will travel deeper into a leaf than will orange, yellow, green, blue, or violet. So what this means is that the chlorophyll molecules can be deeper down in a leaf than they could be if it was trying to do photosynthesis with orange, yellow, blue, or violet light. So the red light molecules, that the chlorophyll, which loves the red light, chose red light nature chose red light to be absorbed for two reasons. Again, one, the fact that some of the peak of the radiation being put out by the sun is in the red region. That's why the sun looks red, orange, yellowish. It looks like that because those are the peak colors that are actually being emitted. And two, because radiation of that wavelength will penetrate deepest in the leaves, so the chlorophyll can be deeper in the leaf if it's absorbing red light to drive the photosynthesis. Okay, now I want you to focus very closely on what the reactants are, where the energy is, and what the products are in the photosynthesis. Now let's turn to animals. Animals do respiration. Respiration is an exothermic process, as we say at the bottom of the slide. Exothermic meaning energy is a product of respiration. So if we pop up here the reaction that animals undergo, you will note that animals, to exist, take glucose, combine it with oxygen, and as a result, they produce carbon dioxide, they produce water, and they produce energy. So the symmetry of respiration and photosynthesis should not be lost upon you. The reactions that plants do to build their structural material are exactly in opposition to the reactions that animals do in order to give them the energy to remain animate. If an animal wants to run around through the forest, it has to take glucose, combine it with oxygen, and it produces CO2 and H2O, but most importantly, it produces energy. And what does it do with that energy? Well, it pumps blood through its system. It moves its mouth and spouts words out that you can hear. It bends its elbow and picks up a glass of beer. So very conveniently, the reactions of photosynthesis and respiration are exactly opposite of one another in every regard. The reactants of photosynthesis are the products of respiration. The products of photosynthesis are the reactants of respiration. Equally importantly, the energetics of the process are exactly reverse. Plants absorb energy from the sun to build the glucose. Animals take energy stored in the glucose and spit it out in order to power their existence. Now, there are still many other things to say with regard to this respiration reaction compared to the photosynthesis reaction as well. First of all, a fact that can't be discounted is the fact that not all this energy really comes from breaking apart the glucose. The energy of respiration also, more importantly, comes from breaking apart O2. O2 is a rich source of energy available to drive processes. It turns out when you put O's together to make an O2 molecule, which is exactly what is present in our atmosphere, we breathe O2 out of our atmosphere, and when we breathe it into our lungs, it is carried by hemoglobin through our blood system, pumped through our cell walls, and then inside our cells somewhere, mitochondria have been fed glucose molecules, and the glucose molecules are ripped apart with the oxygen in the mitochondria to produce CO2, which is carried back away, producing H2O, which is filtered out, and energy. And that energy again powers the human existence, or any animal's existence for that matter. So one more time, I can't overemphasize you the beauty of nature and the synergistic relationship that is painfully obvious from the reactions that make it possible for a plant to exist and the reactions that make it possible for an animal to exist. Okay, so another thing that you should recognize is that not all animals get the glucose which drives their respiratory processes. They don't necessarily have to get it from a plant. So essentially what can happen is, of course, there are certain animals that eat the plants, and then they break the plants apart through respiration and make the energy to make them alive. But some animals eat other animals which have eaten the plants. So you can get glucose from plants directly, of course, but you could also get your glucose supply from a hamburger if you eat the hamburger as well. So in any event, I hope you can recognize now that essentially the carbohydrate known as glucose is of absolute fundamental importance to life, whether it be in the life in the animal form or life in the plant form. Notice that glucose is a so-called carbohydrate. How do I know that for sure? Remember, I said a carbohydrate is C to the N, H2O to the N. So with N equal to 6, I get 6 C's and 6 H2Os, if you like. Now, they're not put together in a way that you recognize them as being individual water molecules connected to carbons, but yet the formula of the carbohydrate still has that basic relationship between the C, H, and O. All right, so we spent a lot of time discussing that. Now let's look at what glucose actually appears to us to be. Now, I'm showing all the connections in glucose. And first, I want to point out that glucose comes in a couple isomers. What did we learn about when we learned about isomers before? Isomers take the same exact chemical formula. That would be C6H12O6. And you just connect the atoms together in different ways. So what we're showing here is that the isomers of glucose both begin with 6-member rings. And the 6-member ring, as you can see, is the hexagon in the middle of both of these isomeric structures. Now, the reason the chemists like to draw it in this fashion, they're showing the thickness coming towards you and then skinnier ones going behind. We're trying to give some three-dimensionality to the appearance of this. So of course, we're drawing this on a flat screen, but we're trying to make it look a little more three-dimensional. So the hexagon ring you see in the middle is trying to give a dimension to it. Now, the other thing you should notice is that the ring has 5 carbons in the ring and an oxygen connected in the ring as well. And this ring is exactly the same in both cases. As a matter of fact, it might be hard for you to discern what is the difference in the two isomers. It's a very subtle and very simple difference. In the so-called alpha glucose, notice the OH on the far right is pointing downward, whereas in the beta glucose, the OH is pointing upward. That's really the entirety of the difference between the two isomers of glucose. Now, the other thing to notice, this is a carbohydrate. It's C6H12O6, but it is certainly not C6H2O6, meaning like the water molecules are uniquely identifiable. The water molecules are spread out in a bunch of different connections than H2O in the glucose molecules. So these are also known as monosaccharides, and maybe that's a name that you have heard at some particular point. A monosaccharide turns out to be a sugar molecule. These are known as sugars as well. and they have a single ring structure. So a monosaccharide is carbons connected in a ring always, and a saccharide, is carbons connected in some kind of ring form that has a carbohydrate CnH2ON appearance, but also there's only a single ring present in the so-called carbohydrate. Now, this is the fuel that drives us and makes it possible for us to be alive. But I will say to you that we really ingest very little glucose directly. Now, I'm not going to say we don't ingest any. We typically ingest glucose, we harvest glucose from other forms of carbohydrates. So when you chew on wheat, it is made of plants that is not really in the form of individual glucoses. It's connected together in what we'll call starch. We break the starch apart. We break the starch into glucoses, and then we use the glucose to run our engine. When we eat sugar, now sugar is also, of course, a saccharide, but it is not the saccharide that you see here. We first break the sugar down. We break it down into individual glucoses, and then we fuel the engine with glucose. You can't run the engine directly on wheat, grain, starch, carbohydrates from that. You can't run the engine on sucrose, table sugar that we normally eat. They first have to be processed to this particular form. Once they are processed to this form, then we can run the engine. So the first thing we have to do is get what we want to use into the form of glucose so that we can perform the respiration reactions that we showed on the previous slide. Okay, now let's just take a short tour of some of the other important carbohydrates that humans ingest, and let's see how they are related and how we can harvest glucose from them. Let's first turn to fructose. Okay, now fructose should have a familiar name to you. This is the form of sweetener that naturally is made in plants. So somehow plants have an enzyme. In photosynthesis, they make this structure, and then plants have some sort of enzymes in them, some sort of factories in them as well, that take the molecules in this form and convert it to this form. Now, what is the main difference between this form and this form? It's the form of the ring. Notice when we go back to the glucose, there is a 6-member ring, 5 carbons and an oxygen. In fructose, there is a 5-member ring, 4 carbons and an oxygen. That is the only difference between glucose and fructose. Now, that doesn't mean anything other than the fact that the ring has been changed. And again, how is this ring altered? Through some enzymatic action of components in the animate tree. Trees are alive, as we well know, and trees have systems not exactly like we have, but yet they do have systems that enable them to process materials, raw materials, and make it so that they exist. So plants take this form of glucose that they get from photosynthesis and convert it to this form of carbohydrate. If you look closely at this carbohydrate, the difference between this and fructose is isomeric, meaning this is exactly the same chemical formula. C6H12O6. So it has exactly the same number of carbons, hydrogens, and oxygens as does glucose. really the main difference, as you can see, is notice how there's like a CH2OH group sticking out of the ring when it's a six-membered ring for glucose. Well, now let's go over to fructose. There are two of those CH2OH groups sticking up out of the ring. That's the source of the other atoms that that make it go from a six-member to a five-member ring. So again, fructose is also a carbohydrate. Fructose is an isomer of glucose. They both have the chemical formula C6H12O6. It's just simply that fructose is a five-member ring compared to glucose's six-member ring. Now again, fructose is naturally sweet. Now I will indicate to you, and we'll show maybe a little more detail on this later. It is not as sweet as table sugar, but of course it is naturally sweet. We ingest it anytime we're eating a fruit, or actually we're ingesting it anytime we eat a vegetable as well. Vegetables have fructose. Of course, vegetables don't have near as much fructose as does fruits. And it is the sweet sensation that you get from fructose when you ingest fruits. All right, so, and again, let me remind you why plants are enticed to make fructose, because they want animals to enjoy chewing on them. Why? Because what they do is among this fructose, they hide their seeds. And then when the animal eats the fruit of the tree or the whatever plant it eats the fruit off of, it's ingesting the seeds of the plant. And then when it walks away from the plant, it deposits the seeds of the plant somewhere far away to spread and proliferate that particular plant species. So this is a cunning system for a tree that can't walk over to the next mountain and plant more trees. So it gets the animal to walk over to the next mountain and plant the tree for it, so that that particular species spreads and thrives. That's the whole secret of what nature is trying to do. Spread, thrive, proliferate. So that's the driving force of doing such things. So fructose is also monosaccharide. Fructose has exactly the same formula, C6H12O6, as does glucose. And again, for the 18th time, I'll say fructose is generated in a plant via processes that the plant has from the glucose that it made from photosynthesis. Okay, so let's now turn to what we call a disaccharide. Now, I think it's not a stretch of the imagination to understand what a disaccharide is. In general, we will classify it as the simplest polysaccharide. A polysaccharide is monosaccharide units linked together. Now, it turns out the linking is always the same, no matter what polysaccharide you have. It's called a glycosidic linkage. And a glycosidic glycosidic linkage. A glycosidic linkage is a O bond between the rings. So a disaccharide has two rings. A trisaccharide would have three rings, and a polysaccharide in general will have multiple rings. But again, what unit connects the rings together? It turns out it is an O bond that does this. Okay, so do I have a picture of one? I don't. I guess I have it on the next slide. So if you take two glucoses and hook them together, you will make something we call maltose. If you take two other forms together of another isomer, so it's another form of glucose known as galactose, and you hook those together, you make a disaccharide known as lactose. So I think on the next couple slides, I will show pictures of some of these disaccharides that are very familiar to us. And it turns out if you take a glucose with a fructose, you will make a sucrose. And sucrose is the one that has become in our favorite form of sweetener that we happen to use. Now, I don't know if you're familiar with this term at all, but in the nutrition world, in the dietary world, of course, we like to put spoonfuls of white powdery sugar on things that we want to sweeten when we're making cookies, or maybe you dump it on your cereal or put it in your coffee. So we put the white powdery stuff that is a disaccharide known as sucrose. But what the food industry has learned is that they can concentrate sweetness and get cheap sweetness by using, there's so much corn produced in this country, they mash it up in various ways. They turn it into alcohol to put into our gasoline, but they also squeeze out of it high fructose corn syrup. I don't know if you've heard the terminology high fructose corn syrup, but this is an intensely concentrated form of the monosaccharide fructose that is squeezed out of corn. And high fructose corn syrup is the general sweetener used for almost everything that we use industrially that tastes sweet. We don't put sucrose on it to sweeten it. We put high fructose corn syrup in it. Like, for instance, Coca-Cola, that has tons of high fructose corn syrup in it, not sugar. Nowadays, though, it's kind of in vogue. You can buy cane sweetened, they call it, cane sweetened Coca-Cola, where they really do use actual sugar in the place of high fructose corn syrup. Now, can you really discern the difference in taste? I guess I don't have a discerning enough palate for me to be able to tell if it's a cane sweetened Coca-Cola compared to a high fructose sweetened Coca-Cola. I can't really discern the difference between the two, but maybe you have the kind of palate that would allow you to be able to tell which one is which. To me, they're both just sweet, so I can't really tell the difference between them. Okay, so let's now just take a look at a quick tour of these various disaccharides and maybe where they are of greatest importance. First, let's start with maltose. And maltose is called malt sugar as well. It turns out that grains and starches have naturally occurring maltose in them. So if you eat the cereals that are not sweetened with high fructose corn syrup, the only sweetness which they will possess would be from maltose. And if you eat your brand cereals and things of that sort, and you know they're not very sweet at all, and many people, of course, they're sweetened in many regards as well, but the only sweetness they do contain would be due to the presence of this maltose, which is a disaccharide that's a combination of two glucose molecules. And this is what it looks like. Now you will notice that it is two glucose rings, 6-member carbon rings. They are connected together. One of the CH2OHs on one and the CH2OH on the other get hooked together, and it makes a so-called glycosidic bond between them. Okay, so as interesting as it sounds, is that not all people have the ability to break these glycosidic bonds. So if you lack the ability to break this bond, then you might have trouble digesting things of this type. So a lot of people have trouble with glycosidic bonds, depending on your metabolic makeup. I mean, I'm not really, again, an expert biochemist or an expert nutritionist, so I don't know the gory details as to what causes certain people to have difficulty breaking this connection. But the point would be this: if you ingest maltose, And now, of course, you want to use the maltose as fuel to your fire. Your mitochondria in your body only know how to process glucose. They don't know how to process maltose. So in order for you to use this as fuel, you have to first convert it into glucose. How do you convert it into glucose? You actually add water across this bond, and it makes 2 units of glucose. But unless you can do that, then you can't process a system like this. So some people have issues with that. Once again, I don't want to make claims about what happens in even something such as a diabetic. I don't know all those sorts of things, but I'm pretty sure it has something to do with the ability to handle things like this. Now, please don't quote me on that. I don't really want to go there because I hate to just make stuff up, but I know that certain people with certain conditions have trouble handling this connection and therefore have trouble processing certain carbohydrates because of this issue. Okay, now if we look at lactose, and I think lactose should be familiar to you as what is known as milk sugar. This is what is present in dairy, and lactose again is any cheese has lactose in it. Many people again are lactose intolerant, and typically what it means again to be lactose intolerant is that you can't handle the glycosidic linkage between the rings in this disaccharide. So again, here is a picture of lactose, and to me, I mean, you have to look really closely to see the difference between maltose and lactose. There's only very slight differences between the two. But again, if you're lactose intolerant, the issue that comes up is the fact that you can't handle this glycosidic linkage, I believe. I believe that's the issue there. And I apologize if anybody out there might have better knowledge about these things than me, but this is one of the problems I believe that people with this issue face. So again, a lactose is a disaccharide. It has a certain level of sweetness, not terribly sweet at all, but certainly not nearly as sweet as sucrose, for sure. And it has this linkage between 2 monosaccharide units, making it a disaccharide. Okay, then this is the one your mom and them are familiar with. Sucrose is a disaccharide. If you harvest it from beets or you harvest it from sugar cane, you'll notice that there's a lot of it present in both of those naturally. And if we just take a look at it, it says that we eat about 100 pounds of it annually. I would say I probably eat more than 100. You can tell by looking at me. I kind of like my sucrose. Okay, so it is a disaccharide, of course, again, and it actually has, very interestingly, this structure. Now, let's compare. I'm going to back up real quick and note that the disaccharide lactose has 2 6-member rings. So it's a disaccharide with 2 monosaccharide units of 6-member ring form each. The disaccharide sucrose has, again, it's a disaccharide with 2 rings, but notice it has a fructose hooked to a glucose. So this is C12H22O11 is the form of this particular disaccharide. There's an H loss. There's some, when you make the glycosidic linkage between the 2 rings, you lose some of the water from it. So instead of being C12H24O12, you lose a water molecule when you link these together, and you get C12H22O11. So it's lost one of the water molecules in the connection to make the disaccharide. Okay, now let's then, since this is our main thought so far with our discussion of carbohydrates, let's mention a little bit about sweeteners. And first of all, you should stop to think about this. What does it mean to be sweet? Well, what it means to be sweet is that we have certain receptors, is what we call them. And what a receptor is a particularly shaped organic, huge organic molecule. And the shape of it is absolutely critical to its behavior. So it's a very large molecule that a sweet molecule fits into the structure. So this huge molecule has certain pockets to it. And the pocket is the perfect size to fit another molecule. And if the molecule fits in there perfectly, it stimulates electrical impulse to send the sensation through your nerves that tells you, ooh, that's sweet. Now, if you ingest this and it fits into these receptors, but doesn't fit in there nice and tight, like a key that can open the lock, then you don't send out the impulse or signal that it is as sweet. So these certain molecules, these fructoses and glucoses, maltose, lactose, and sucrose, have, with their disaccharide structures, a certain shape that when they slide into a so-called receptor, trigger the response of neural stimulation and endorphin release in our bodies that send out the signal, ooh, that tastes sweet, or that's not as sweet, through some very complex biochemical signaling that goes on in the body. So sweeteners are naturally occurring or man-made agents which either naturally or artificially enhance flavor. And of course, mono and disaccharides are naturally sweeteners, but of course, humans over the past 50 years, I would say it's been maybe even more, oh, certainly more, we've been making artificial sweeteners. I think saccharin was the first artificial sweetener, probably made in the 1940s, which is, it's going on 80 years ago now. So we've been synthesizing sweeteners now for going on 80 years, as well as these naturally occurring synthesizers, or naturally occurring non-synthetic sweeteners that we've been harvesting from plants, sugarcane, beets, and things of that sort, and high fructose corn syrup. We've been harvesting these things for quite some time now as well. So this is an artificial sweetener. Again, as I point out, this is one of the first ones. You'll notice that it is also a 6-member ring hooked to a 5-member ring, but it's got quite a different structure. First of all, I want to point out to you, I drew this myself, and I didn't put all the H's on. So there are a bunch of H's on this, in addition to the atoms that I've shown. And you'll notice I left some of the points blank. There's connections that meet at a point, but there's no atoms listed. Anywhere where the atoms meet at a point and there's no atoms listed, you put a carbon, because I got lazy. So there are where you see connections but no symbol, just replace that with a carbon. And generally the carbons are attached to hydrogens as well. So I left off a bunch of hydrogens on this also. What you should notice though is that saccharin is a 5-member ring attached to a 6-member ring. So in some way it mimics sucrose. Remember, sucrose is a 5-member ring attached to a 6-member ring. But in sucrose, there's this so-called glycosidic connection between the two rings. Saccharin does not have that. Now, saccharin does have oxygens, as you see, but in addition, you can also see saccharin has nitrogen, saccharin has sulfur. Now, it turns out, though, that when you ingest this molecule and you chew it up in your mouth it slides into the keyhole in much the same form that sucrose slides into the keyhole. And when it does so, then it fits the keyhole, triggering the response, ooh, that's sweet. So this is shaped particular. If you look as particular at how its electrons are distributed, the distribution of its electrons kind of fool the receptor into thinking that it's got the same type of response to it that would come from, say, a sucrose molecule. So why would we want to do something like this as opposed to using a sucrose itself? Well, it's because of the fact that now when we ingest too much carbohydrate, we haven't discussed what happens when you ingest too many carbohydrates. What happens when we ingest too many carbohydrates is that humans have a built-in primal response. Let me explain it to you in this fashion. Thousands, tens of thousands, maybe 100,000 years ago, we weren't so lucky as to encounter food very often. Or we probably did okay because we made it these 100,000 years. But believe me, it was an issue when you could encounter food. So what advanced animal systems learned to do was, if you encountered an excess amount of food, store it in your body for later use. So if you ran into too much carbohydrate, you converted the carbohydrate into a storable form that you could put on or keep on you, and then instead of just crapping it out. So generally you could crap it out, but the other thing to do was change it into another form so that it could be stored. And then if you came across a period where you didn't run into any carbohydrates, you could convert it into the usable form. So we developed what we know as fat. Fat is a storable form of carbohydrate. And 100,000 years ago, it was important that you could store this fat because there was guaranteed going to be a point where you might need to go to the reserve and use that fat in order to sustain your life. Well, guess what's happened? We don't have that problem anymore. We got plenty of carbohydrate around. So we ingest plenty of carbohydrate. But the problem is, nobody turned off this storage system. The storage system has been here for 100,000 years, and it'll be here probably for the, well, I can't say for sure, but it'll be here for a long time. So your body still has the primal instinct to store this excess carbohydrate in case you need it later. We don't need it later. So what happens? We just keep storing it and storing it and storing it. So that ain't good. So what does an artificial sweetener do? It's not a carbohydrate. So your body doesn't say, ooh, let me store this carbohydrate for later. So when you ingest an artificial sweetener, if it is not used, well, okay, it gives you sensation in your mouth and all that other stuff, but then it is not converted into a storable form. It's just crapped out. So that's the benefit of using an artificial sweetener compared to using these carbohydrate forms of sweeteners. We don't have the mechanism to turn them into fat and keep them around for a rainy day, which unfortunately we don't run into many rainy days anymore. So we're just carrying this stuff around with us. Okay, so I'm just showing you here a couple other types of sweeteners that are also very popular. Now this one doesn't look anything like a 6-member ring and a 5-member ring. It's called aspartame, and aspartame goes by the trade name NutraSweet. And this is something called a cyclamate. And this is actually a sodium salt of a cyclamate. Cyclamates are, notice it does have a 6-member ring. So for some, you can understand why that might stimulate the response that a natural sweetener would stimulate. So again, the benefit of ingesting these is you get the sensation of sweet, but the body doesn't say, hey, this is a carbohydrate that I can store for later and maybe turn into glucose down the road if I can't run into any glucose. Okay, so this is kind of telling a little bit about the story, what we mean by sweet. So they correctly bind into a certain receptor that stimulates and triggers this. So the binding mechanism depends on the shape and how electrons are distributed around the sweetener. So this is my cartoon trying to show you what is going on. The bigger, lighter blue molecule is the so-called receptor. And then if a molecule binds to the receptor properly, it stimulates some neurological response. So it will stimulate, for instance, the sensation of taste that tells you that that is a pleasant taste that I'm deriving from that. So the shape and charge of the sweetener specifically tip off the receptor biomolecule and trigger all kind of bioactivity, like, again, endorphin release, pleasure release. I mean, all kinds of responses come from these types of interactions. Now, what I'm showing here are, this is a sucrose molecule that, and it looks a little bit weird the way I'm drawing it here, but this has a 6-member ring and a 5-member ring that chemists use in a modeling program to show what they look like. Now, if you put the charge on top of this, this is just the nuclear skeleton. Let me now put the charge over the top of this thing. And when I do that, there it is. This is what the charge looks like when you spread it over there. Now, the code that this is using is where you see the red spots. This is where there's lots of electrons. Where you see the blue spots, there's where you see lots of positive charge. Green means electrically neutral. So again, red means negative charge, blue means positive charge, green means no charge. So here's what I'm saying. This molecule will come floating through space into a huge receptor molecule. It will bind into the receptor molecule. And if the receptor molecule feels the red spots where they are, feels the blue spots where they are, and the rest, the green spots, meaning there's kind of regions of high negative charge, regions of high positive charge, and then electrically neutral regions of the molecule, it stimulates in the receptor. Ooh, that's sweet. So this is what the electron distribution around sucrose looks like. It's also possible to draw the electron distribution around the other things. Here they are. Okay, there's sucrose, there's fructose, there's glucose, maltose, saccharin, and cyclamate. And down here I got my aspartame, and down here I got ethylene glycol. Now, let me just pop up what these maps look like on all of these. You can see that many of them have similarities. Let me back that up. Notice the top left corner is what a sucrose looks like in terms of, again, it's negative, positive, and neutral spots. Fructose has similar negative, positive, and neutral spots. Glucose, negative, you see the negative, positive, and neutral. And then when you look at all the other sugars, you can understand why maybe when you chew them up and they fit into these receptors, that they might have similar characteristics. Now, the one on the bottom right corner is ethylene glycol. This is the main ingredient of antifreeze. I don't know if you've heard the story about antifreeze, but if you ingest it, in particular, dogs might drink it, it's sweet. So antifreeze tastes sweet because the molecule that's used in antifreeze, as you can see, has many of the characteristics of the way charge is distributed around it, just like sweeteners do. So ethylene glycol or antifreeze molecules can bind into the receptors of a dog's mouth that say, "Ooh, that tastes good." So the dog drinks more ethylene glycol and ends up not in good shape as a result of that. Okay, I think that would be it for today. Remember, please go to Moodle. Do test 3, part 2. It will close Monday at 11:30. So make sure you go there and do that. So until next time, next Monday, peace out. Thanks.